Well, thank you for this introduction, and of course, I would like to thank the two organizers for having given me the opportunity to present our work on this special relationship that developed between African trypanosomes and humans. I guess you all know that African trypanosomes are unicellular organisms, unicellular parasites that are able to infect a wide variety of mammalian hosts. And what you should keep in mind is that these parasites are continuously extracellular, so they never enter the cells of the host. You actually see here a Trypanosoma brucei parasite, which is a prototype of African trypanosome, happily swimming in the vessels of liver of an infected mice, and you immediately see that the parasite is to deal with the cells of the immune system of the host, and this is, of course, the topic of my presentation. So the most characteristic feature of the interactions that take place between these African trypanosomes, particularly and mammals, including humans, is the fact that these parasites are fully covered by a very dense coat made of something like 10 million identical molecules, glycoproteins that are called variant surface glycoproteins, or in short, VSG. And this coat is so densely organized that only the loops at the very surface of this coat are accessible to antibodies. And actually, the VSG, each VSG, again, there is a single type of VSG that is covering the full surface of the parasite at any given time. This VSG is made of two domains, the N-terminal one being made of an hairpin-like structure of two long amphipathic alpha helices, helix A and helix B, helix A being at the N-terminus of the protein, and then there is, of course, a short turn at the bottom of the structure, and then you have helix B, which is an helix which interacts with helix A, as you see here. So altogether, this makes an elongated structure that you see here, and that presents the antigenic surface loops to the host. This is important to remember for the following of the talk. Now, the reason for mentioning the VSG is that it's basically an antigen, so presenting epitopes at the surface to the host. And what is striking in the case of the VSG is that this antigen is changed every week. So what is required for the parasite to stay in the host is to regularly change at least the surface exposed loops that are playing with the antibodies. And the reason for that is that doing so, the parasite managed to this allows the parasite to stay for a long period of time in the blood, because its parasite is playing with the antibody response of the host to continuously control its own growth. You can easily imagine that if there was no control of the parasite growth, then the host would be too quickly destroyed, and of course it's not the interest of the parasite that the host dies too soon. And this system was developed because, in particular for African trypanosomes, the transmission of the parasite from one given host to the next is very, very inefficient. As you probably know, that this is through the tsetse fly, and this is a system that is not good, not very good. So the parasite has to develop long-lasting chronic infection in order to survive. So this is a system that managed both the host and the parasite to survive for a long period of time, weeks, months, or even years. Well, there is another characteristics of the interaction that takes place between the African trypanosomes and the immune system of the host is this special system of resistance of humans in particular, as you see this sophisticated scheme. So it means that the prototype of African trypanosome that I've already mentioned, TB brucei, Trypanosoma brucei, is able to infect, as I said, a wide variety of mammalian hosts like cattle, where the parasite causes a disease which is called Nagana. But specifically in humans, the parasite cannot infect these individuals. And this is also true for gorillas. So in the case of humans and gorillas in particular, and specifically, there is something in the blood that was called the trypanolytic factor, or in short TLF, that protects us very, very efficiently against infection by the parasite. So it's a special system of innate immunity that protects us against this given parasite. And of course, the reason why some parasites, some African trypanosomes can infect humans, and you probably know that where these parasites cause a disease called sleeping sickness, is simply due to the fact that two individuals from the prototype TB brucei have evolved to resist TLF. Okay? So these two are almost certainly clones, and as far as we know, there are only two such clones that found the ways to escape TLF inactivation, or destruction by TLF. So this is important to keep in mind. Naturally, we are protected against the parasite, and then if people are suffering sleeping sickness in Africa, it's just because two clones managed to resist our natural defense system. So in this talk, what I would like to do is to tell you the story of the successive interactions that took place between African trypanosomes and humans in particular. But don't forget, when I say humans, it's also true for gorillas. So it only appears very recently during evolution, but it's not just humans. So the ancestors of gorilla and humans have built a specific system of innate immunity, as I just mentioned. What is it? So to make a long story short, in 2003, we proposed that the factor that in our blood kills African trypanosomes is a given protein named apolipoprotein L1, or in short, ApoL1. I will briefly allude later on in the talk on the way we found this. But yes, ApoL1 was a protein already known to bind lipid particles, and in particular heavy density lipoproteins, or HDLs, and even the densest fraction of HDL, which is called HDL3, and which is usually named the good cholesterol. So in these particles, ApoL1 is a minor component, and you shouldn't be confused with apolipoprotein A1, which is the major components of these particles. So ApoL1 happens to be a minor component of HDL3 particles. And again, it's important to keep in mind, so ApoL1 is a human-specific protein, human and gorilla-specific protein. So we proposed that that protein was the lytic factor for several reasons, as you will see. But at the start, we were confronted with a controversy because other people in other labs have published before us that the trypanolytic factor was another protein, another human-specific protein called haptoglobin-related protein, or in short HPR. And again, like for ApoL1, the function of this protein was not known. But in that case, you could guess, because haptoglobin-related proteins, as the name tells, is very, very similar to haptoglobin. And you know that haptoglobin is a major component, or inflammatory component, that binds hemoglobin. Well, the same for HPR. HPR exists as a complex with hemoglobin. So what is amazing is that HPR, or the HPR-HB complex, is also bound to the same HDL3 particles that also contain ApoL1. So we have here a very strange situation, which is the presence on the very same particles of two human-specific proteins that were both evoked or presented as involved in the defense against trypanosomes. So obviously we had to work on that because there was a problem. So at the end of the day, to our full surprise, we realized that actually African trypanosomes possess a given receptor for the HP-HB complex, haptoglobin-hemoglobin complex. And the reason of the presence of this receptor in trypanosomes is simply the fact that it's good for the parasite to have the receptor. Why? Because this allows the parasite to internalize the complex between haptoglobin and hemoglobin, and trypanosomes like to have heme from hemoglobin. Yes? They don't care about both HP and HB, they readily destroy it, but they keep heme that is present in hemoglobin, and this allows the parasite to somehow resist the oxidative response of the host, and it's also a growth factor for the parasite. So it was a good idea for TB brucei to have this kind of receptor at the cell surface, but as you can imagine, in human blood, or gorilla blood in particular, it becomes a problem, really, because, as a matter of fact, this receptor cannot discriminate at all between HPR-HB and HP-HB, okay? So it means that in human serum, the presence of this receptor on the surface of the parasite is the best way to catch and internalize toxic particles that will ultimately kill it, okay? That's the way it is. So through the binding to this surface receptor, the particles that do contain the toxin ApoL1 are internalized through the mouth of the parasite, which is called the flagellar pocket here, and then ApoL1 can efficiently enter the digestive system of the parasite and ultimately kill the parasite. I find it a very clever way to kill the parasite, and we were inventors of that, you know, by having particles containing the toxin and then the key that allows the efficient uptake of the toxin into the parasite. At the same time, this is a way to explain the controversy, because ApoL1 is indeed, as we said, the toxic factor that kills trypanosomes, truly, but HPR has a role to play in this system because HPR is involved in the efficient uptake of the toxin within the parasite. So what is ApoL1? As I said, this protein was only known by the fact that it's bound to HDL particles, and so we had to do everything from scratch here, and we realized that this protein contains ionic pore-forming domain, which is relatively similar to a known ionic pore-forming domain of some bacterial colicins. And in the case of ApoL1, this pore-forming domain allows the passage of chloride ions through membranes. So, in other words, ApoL1 can be inserted into a membrane of any kind and organize the passage of ions, or in particular anions, through this membrane. And the way ApoL1 actually kills the trypanosome in vivo is summarized in this slide. So, this is the mouth of the parasite, so the flagellar pocket. The particles, HDL particles, enter the parasite through the binding to the HB receptor. These particles are then taken up inside, and upon acidification that occurs during endocytosis, ApoL1 is released from the particles and inserted into the next possible membrane, which is the endosomal membrane. And flux of chloride that is triggered by the presence of this ApoL1 in the membrane leads to considerable swelling of endosomal vesicles and ultimately the lysosome, which is really considerably swollen at the end of this process. But despite the spectacular swelling of the lysosome, this is definitely not what kills the parasite. What kills the parasite is the fact that due to the activity of a given kinesin, which is called TBQC1, some membranes that contain ApoL1 are transferred from endosomes to the surface of the mitochondrion, and this results in a kind of apoptosis of the trypanosome with the formation of megapores that allow the release of a given endonuclease called TB-endo-G from the mitochondrial membrane to the nucleus, where this endonuclease cleaves the DNA simply, and then, of course, the parasite cannot survive anymore. So this is the way ApoL1 kills the parasite. Now, I've just described step 1, and then we enter step 2, which is the parasite response. Okay? And again, this is something that I've already mentioned at the beginning. 2 clones managed to do it, despite the sophistication of the system, again, with a toxin and a key to enter the parasite. 2 individuals managed to find a way, which is remarkable, yes? And so nowadays, these 2 are responsible for trypanosome infection in Africa in the field. Let's start with TB rhodesiense, which is typically found in Eastern Africa nowadays. Because that's where the story started in 1998, as you can see. At that time, we were working on the mechanisms of antigenic variation. And regarding TB rhodesiense, we were struck by the fact that these mechanisms of antigenic variation are actually linked to the way this parasite can resist TLF, or by the way TB rhodesiense can infect humans, okay, or to be infective in human serum in the lab. Okay, so let's be clear that this parasite is not constitutively resistant to TLF. In vitro, you can readily lyse the parasite if you put it in human serum. It can acquire the ability to resist TLF, so it's inducible. And in order to understand the mechanism which is at play here, I have just to give you a few words about the mechanisms of antigenic variation, very, very, very briefly, just enough to understand here. Actually, for the VSG to be expressed on the surface of the parasite, you have, of course, to transcribe a VSG gene. The parasite possesses a lot of VSG genes, but as a strict, absolutely strict rule, a single VSG is expressed at any given time, so that every given parasite has only a single type of VSG, 10 million molecules of VSG, but the same, so it's homogeneous on the surface. Well, again, to be transcribed, the VSG has to be in a given special genomic location, which is a telomeric location, and there are 20, something like 20 sites called VSG expression sites where the VSG can be transcribed. To summarize, you have 20 possible VSG expression sites or telomeric, but as a rule, a strict rule, only one of these different VSG expression sites is transcribed at any given time. But this can change. So an active site can become silent, and another one active instead. This is a process, this is one of the mechanisms of antigenic variation, because switching from a given expression site to another leads to the change of the type of VSG expressed on the surface. That's one thing. The second feature that we have to know is that in these telomeric VSG expression sites, you have a polycistronic transcription unit, always. So these sites not only contain the VSG gene itself, which is the last one close to the telomeric repeats, but you have also a collection of additional genes as well that are all transcribed by the same RNA polymerase molecule. So to put this together, we could understand how TB rhodesiense depends on antigenic variation or is related to antigenic variation for expression of resistance to TLF, because we found that for rhodesiense to become resistant to TLF, it is forced to use a single unique VSG expression site, always the same, from the collection of 20 possible expression sites. So that one, we called it resistance, the expression sites for resistance to human blood or to TLF. And we immediately understood why. It's because this particular expression site contains a given gene here called serum resistance associated gene that is the transcription of which is absolutely required to resist TLF. And the experiment that led us to understand this was absolutely simple and straightforward. We took TB brucei, so the prototype of African trypanosome, which, as I said, is fully sensitive to human serum, and we just introduced SRA into this parasite. And doing so, we reproduced what was done by Mother Nature long before us, which is transforming a non-pathogenic parasite into TB rhodesiense. So, and as far as we know, it's correct to say that TB rhodesiense is nothing else than SRA expressing TB brucei. What is SRA then, which is able to confer protection on its own? Well, amazingly, it's very similar to a VSG. Actually, it was a VSG before a deletion occurred at the tip of the structure here, because as a matter of fact, SRA is nothing else than a defective VSG. So it's a VSG that lacks a sequence here, and very strikingly, the sequence that is missing is precisely the sequence that exposed the surface epitopes. So it's very strange to realize that the factor that confers resistance to TLF is an antigen without antigenic loops. Okay? So it's clearly a defective VSG. So to understand how this could confer resistance to human serum, we, at the end of the day, realized that this SRA protein is able to interact strongly with ApoL1. And by the way, this is how we discovered ApoL1. Okay? It's through affinity chromatography using SRA that we catched ApoL1 from the blood, and we realized that SRA strongly binds to ApoL1, and more particularly, it's the helix A of SRA, so the N-terminal helix that I shown you at the beginning of my talk, and there is a similar helix in the C-terminus of ApoL1, and this is a strong interaction, coil-coil interaction between these two helices that allows SRA to inhibit the toxic activity of ApoL1. Interestingly, at least in my eyes, the fact that SRA is depleted of surface loops is because it's necessary for this protein to reach ApoL1. Okay? If you had a normal VSGs go to the surface. They play with antibodies. They are targeted to the plasma membrane. So SRA, if you make it again a good protein, a good decent VSG, by putting back the surface loops, we managed to find the gene from which SRA has evolved by deletion, yes? So we could put back the missing loops in SRA. Then you lose the ability of SRA to confer resistance to TLF, simply because then SRA goes to the surface and not in the endocytic system. And actually to find ApoL1 and neutralize ApoL1 directly by direct interaction, of course, you have to go to the endocytic system. And in trypanosomes, it's known that defective VSGs are targeted by default to the endocytic pathway for degradation. So this very system was used to resist the toxin that was made by the host. Personally, I found it remarkable, but you may think otherwise. So this is it for TB rhodesiense, and let's go to TB gambiense now. How does it resist TLF? Long and very difficult work. It took us a long time to solve this question, but at the end of the day, we managed to do it. And you see here the model of the two helices of TGSG. The protein is a VSG-like protein again, like SRA. It's a VSG-like because it's a VSG, but which is devoid of the C-terminal domain. So something is missing too in this case. Well, the protein has been called TGSGP for Trypanosoma gambiense specific glycoprotein. And we could show that what is necessary for providing the resistance to TLF in the case of TGSGP is it's a peptide which is in between the two helices that you don't see on the model because we couldn't model this peptide. And what is important here is a beta sheet that we have shown to be able to confer, to give stiffness to membranes, experimental membranes. And the ability to stiffen membranes by this peptide and by TGSGP as a whole is strictly linked to the ability to confer resistance to TLF. So we have proposed that in this case, the resistance is completely different from the one used by TB rhodesiense. This time, instead of neutralizing the toxin, the parasite protects himself against the toxin. So the membrane become now resistant to insertion by ApoL1. This is summarized in this slide in order to be as clear as possible. So the template that was used to make adaptive proteins in both cases is the same, is the VSG. In the case of rhodesiense, it's the helix A that was used to neutralize ApoL1 by direct interaction. And so you see here ApoL1 entering the endocytic system from the flagellar pocket to the lysosome. And in its journey in the endocytic system, ApoL1 interacts with SRA so that the complex becomes inactive. In case of TB gambiense, this peptide here, which remember is at the very bottom of the structure because there is nothing underneath, so there is no C-terminal domain. This peptide is in close contact with membranes, and it's able to organize kinds of carpets on the surface of the membrane, stiffening the membrane, and so that ApoL1, when entering the endocytic system, cannot insert anymore into the membrane of the parasite. So these are two different ways for the parasite to resist the same ApoL1 toxin. You may think that this was the end of the story, but amazingly we found that it was not, you know, because when working on the system, we went to discover that there was a step 3, which is the fact that despite the mechanism developed by TB rhodesiense, humans took over in the field. And so we found that in the field in Africa, many people, many humans developed resistance to TB rhodesiense. And the way this was seen is because there is a price to pay for that. People resisting rhodesiense have a strongly increased risk of developing chronic kidney disease, so kidney insufficiency. Well, this part, so step 3, started in my lab in 2009, when we were trying to develop ApoL1 mutants able to kill rhodesiense now, become, after mutation, experimental mutations, they would be resistant to neutralization by SRA. So we knew that the parasite antidote, SRA, was interacting with the C-terminal hairpin of ApoL1 through coil-coil interaction. So it was relatively easy for us to develop, by side-directed mutagenesis, an altered helix in the case of ApoL1 that wouldn't be any longer able to be neutralized by SRA, okay? And if the mutation would preserve the toxic activity of ApoL1, then we would make drugs active again against this human pathogen, TB rhodesiense. And it worked. So we made several mutants of ApoL1, recombinant ApoL1, that were perfectly able to kill TB rhodesiense. Then we entered immediately in collaboration with American nephrologists because, believe me or not, but the mutations that we did in the lab somehow already existed in the field. Not exactly the same, very close mutations, but the result is exactly the same. So people in the nephrology field were looking for SNPs that would be linked to kidney disease, chronic kidney disease, and they were puzzling because they couldn't, you know, choose between the different possibilities in a given locus. But after our publication, they realized that the mutation that we did in the lab were so close to some of the mutations that they found very strictly linked to chronic kidney disease. So these mutations are called G1 and G2. And so since the discovery in 2010, this has been fully confirmed. Actually, now we are pretty sure that these mutants, ApoL1 mutants, induce kidney disease. So you see a situation, the mutations are meant to protect against infection by TB rhodesiense. These mutations are widespread where? They are widespread in Western Africa, where rhodesiense doesn't exist nowadays. And I'm ready to bet that the spreading of these mutations in Eastern Africa led to, in Western Africa, led to the disappearance of TB rhodesiense from this part of the continent, where it has been replaced by TB gambiense that uses another system of resistance to ApoL1. So in my opinion, two important points here. So despite the fact that these mutations have been are toxic because they cause kidney disease, they've been selected by evolution, and meaning that it was probably more important to resist infection by African trypanosomes for a human to survive. And remember, humans did appear in Africa indeed. And this adds to the fact that, as I mentioned at the beginning, we have a special system of innate immunity against this parasite, also pointing to the fact that it was important to find a way to solve the problem of this endogenous parasite in Africa. Alors, to finish, what is ApoL1 at the end of the day, or rather, what is ApoL? because ApoL1 is just the last member, most recent member of a large family of proteins, ApoLs, which are present in all mammalian organisms, a very dynamic family of proteins, the function of which is absolutely unknown. And so we started, the ApoL1 in particular is the most recent member, but it's also something special among ApoLs because it's the only one which is provided with signal peptides that allows the protein to be secreted and present in the serum where it can meet ApoL1. All of the ApoLs known are intracellular, so the function, original function of ApoLs is almost certainly intracellular. So we have worked on ApoLs in mice, and we found that typically the expression of these proteins is in antigen-presenting cells, dendritic cells in particular. We have particularly worked in dendritic cells in mice and in humans as well, and also in kidney podocyte, which is of course relevant to the problem of the disease. Kidney podocytes are actually professional antigen-presenting cells as well, okay? And what triggers the expression of ApoLs in particular, regarding especially ApoL1 and some others ApoLs, is viral activation of the TLR3 pathway. So this strongly induces ApoLs. So ApoLs belong to the inflammatory response. And what we have found is that in this response, ApoLs are particularly involved in the cellular death that is triggered upon activation of dendritic cells. So, of course, when there is activation of antigen-presenting cells, one of the facets of the phenotype is to die as soon as the work has been done to prevent excessive inflammation, etc. So, apparently, ApoLs are involved in this process. So, they are induced for what? To stop the lifespan of activated antigen-presenting cells. So in my opinion, in the case of ApoL1 in particular, the toxic potential, so to trigger cell death of ApoLs, was used to work out of the cell as regard to what it was doing inside cells. Perspectives of this work, of course, there are many, but the most important are, of course, that the attempts of raising cattle that would resist African trypanosomes. So this is currently underway in Scotland. So cattle expressing ApoL1, or rather ApoL1 variants. In my lab, we have an ApoL1 variant, experimental ApoL1 variant, that is able to kill absolutely all African trypanosomes, whether they are pathogenic for man or for animals or whatever. So we have an ApoL1 variant which is able to kill all African trypanosomes whatsoever. You can normally, this should be possible to inject recombinant ApoL1 mutants because to treat people suffering sleeping sickness, because the activity of this protein is incredibly strong. So you don't need lots of protein to achieve the goal. And last but not least, and we are actively on that, the fact that ApoL1 mutants are involved in kidney disease opens ways to try to understand the mechanism of kidney insufficiency. And believe me, we have found very, very interesting things, unexpected things regarding this, but maybe next time we will be able to present this. But so this is, of course, related to the natural functions of ApoLs in the podocyte and kidney physiology. And lastly, let me list the names of the principal investigators in my lab who have been involved in this saga. And of course, I thank you for your attention.